drug targets. Now, RNA-seq, which is quite common, so there is nothing much to describe. And if you want to study a full-fledged, detailed comparative analysis between spatial transcriptomics and normal NGS, next-gen sequencing, then this is one of the reference paper for which I have included the screenshot, and this is the link. So this will give you the detailed insight, but I will always recommend to read the research and review papers of the main developer who are from KTH, and they have published a really large number of high impact factor papers on different applications, and most of these applications have been published by the developer of spatial transcriptomics techniques. Now, if you want to understand the application of ST or spatial transcriptomics, it could be applied for metabolic programming, dynamic gene expression, signaling pathways, activation, cell-cell interactions, spatial expression patterns of specific genes, a spatial distribution of different cell types, CTCs, CSCs, LSCs, for example, in case of immune programs, a spatial regulation of immune surveillance and immune escape. Now, this could be, in terms of application, it could look like, it could go for target discovery, disease understanding, personalized medicine, therapeutic responses, drug action network, pathway prediction, pharmacological model, drug discovery, and many more applications. Now, the chip of a spatial transcriptomics or genomics could look like somehow this. So this is how it could look like. All these red dots means these are kind of control where there is no labeling, and each red dot represents a single barcode, and each barcode could map to approximately 200 cells and their genes. So if you want to go for look over application, for example, these are the prostate biopsy sample sections. These are the 4 sections out of 12 sections. And if you place these biopsy slides on the chip, it could look like this. And then for all these slices, you could see here. And this is the heat map for gene expression on the for each barcode in different regions. For example, if you go after normalization, this could look like this. So this is like exactly kind of tissue, how the tissue look like. Red means the expression level is quite high. Blue means the expression level is comparatively down. So this is section 1, 2, section 1 to this one, and then section 2, 1, this one, section 3, 1, this one, section 4 to this one. And we have done, like I also was involved in this technique. So we have also tried to go for some kind of prediction of which region is like tumor or cancer region, which region are normal region, and what are the adjacent tumors or adjacent tissues. So for example, this is trunk mutation, these are internal branch, and these are the branch, a specific branch. So there comes the terms like passenger and driver mutations, which I will be discussing in the next slides. Now, you could use this technique to classify cell types within a tumor-based phenotypic gene expression, determine intratumor heterogeneity, cancer evolution, and which cellular pathways are altered between cooperating cell types, determine how different cell types within tumor tissues responds to anti-cancer target treatments, which cellular phenotypes or genotypes correlate with response or resistance, and thus develop an assay to assess intratumor heterogeneity or intertumor heterogeneity. This is one of the paper which was published in New England Journal of Medicine and by Garlinger. So it is one of the best thing to understand how to classify different regions of tumor. And these R1, R2 represents tumor. So they have not used here the spatial genomics or transcriptomics concept, but they try to use maximum, they try to maximum use the next gen sequencing approach to go for a spatial understanding of genes and mutations, for which they have divided the tissues into different regions. So the tumor samples were sectioned into different regions, and these regions are labeled as R1, R2, and so on. And they have also plotted like the mutations, then they have plotted the phylogenetic relationship, how they could look like to identify different types of mutations. And then they go like for final conclusion. This is the very important paper which you could look and read the entire paper. And I think it is quite easy to access this paper also. So if you try to identify and go for more functional impacts analysis, you could use some of the similar techniques or approaches for analysis of a spatial transcriptomics or a spatial genomics data. For example, gene expression data analysis, DIX analysis, differentially expressed genes analysis, then clustering to identify different clusters of cells and genes, and then go for pathway analysis, and finally try to also understand the pathway interaction or pathway crosstalks. Then there are many more applications which you could do by using these two techniques, including spatial transcriptomics. For example, mutational processes, identify the mutational signatures, and these things have been described in detail in Nature Review Genetics. I think it was 2013 paper and a 2014 paper. Yes, and this is the same thing. If you want to understand the mutational signatures, they're like three code, bases, uh, mutational signatures. Then look for something like whole genome sequencing for cancers in different patients, then mathematical extraction of mutational signatures from aggregated data, then go for dominated CTG like C2T, and then C2A, C2T, C2G, and then something so on. And these, there are a number of papers available from this group and their collaborators also from UK Nick Journal. Then next part is like, this is the real application and analysis which I have done for all the 12 sections. For example, how many total number of mutations you could analyze for each sample, all the 12 samples. If you could see, the highest mutation is on sample number 4, sample number 7, then so on. This is how you could look, and the same thing have been plotted in pie chart. Now, if you want to understand the mutation, total mutations and their distributions on different chromosomes. For example, chromosome number 1 is showing quite high number of mutations in most of the sections, and all these different colors are representing different chromosomes and the total number of mutations. The bigger the size of this means the bigger number of mutations are present in different sections on different chromosomes. So now we could also go for finally clustering to identify the clusters of cells or cell groups, because each barcode has 200 cells. Then let's say we took top 5 clusters, then we went for the differential gene expression analysis, number of mutations, and so on. So in this way, you could analyze the gene expression, differential gene expression in different regions, and the pathway enrichment analysis in different regions also. Now, for example, this is the detailed analysis for the same 12 sections of spatial genomics. Then what type of mutations are dominantly present? For example, T2C and A to G are almost high in all the samples. But if you compare, they are somehow much higher in four samples. For example, S4, S2, S7, and S3. And then it comes S8, S9, S12, S6. And then there are four samples which are showing quite suppressed lower number of mutations in almost all types of mutation and for all the chromosomes. And after detailed analysis, we identified that these four samples are completely cancer samples or tumor samples, and these four samples are completely healthy, like S1, S5, S10, S11. And these four samples are adjacent tissues S6, S8, S9, and S12. They are adjacent tissues. So this is how you could classify. Then it comes, you could also identify what are the passenger mutations and what are the driver mutations for cancers. So if you want to identify the driver mutations, these mutations will be the main driver mutations. That means these mutations are forcing the cells, normal cells, to turn into tumor cells. So this is how you could identify. And this is like trunk, or we call it passenger mutations. That means these are not specific to one, but we took the common mutations so that to understand what are common mutations between all the 12 samples. And then we identified what are the main difference, and that is what helps to identify driver and passenger mutations. So you could see here the trunk, internal branches, and main branches, and then the driver and passenger mutations. So T for tumor, AT for adjacent tumors, N for normal samples. So this is one of the best example, and this is quite unique. So please keep in your mind and look over these lectures again and again to understand the new techniques which most of the people are using these days to their research projects. And then that is overall what you could think about. So please subscribe this channel if you want to learn the relevant and new advancement and their applications in bioinformatics and clinical research, diagnostic research, plant biology, animal biology, and many more things. So please subscribe this channel. Thank you very much.